This lecture is going to explain you about skeletal muscle tissue. So it is the tissue which makes skeletal muscle. So the muscles we see on our body, which we can touch, it is a organ. So let's say if we touch our biceps muscle, it is a skeletal muscle organ. So what we are trying to understand today is the tissue which make these muscles. Let's go to the next slide. I believe everyone remember this gentleman. He is still alive. Look at him. He got lots of big muscles. How did he get it? How did he develop it? What are they made of? Before I talk about this lecture, I was looking on the website trying to find who identified or who starts talking about these muscles and their microscopic structures. I saw 4 persons, but I could only get 1 person's photograph. The first 2 persons, Draper and Hodges, they are from Australia. And second 2 scientists, Hansen and Huxley, they are from England. So with the development of electron microscope, they were able to identify these microscopic structure of muscles, which you are going to learn in later part of the lecture. So it was around end of 1940s, early 1950s. They could identify the microscopic arrangement of the muscles. Before I go further, I want to remind you this slide. So this is a slide we all seen in our previous lecture. A human being is made of many different types of cells. A cell is a structural unit of living, and when these cells with the same intention and same manner group together, it becomes tissue. Generally, we have 4 tissues types in our body. They are number 1, epithelial tissue, number 2, connective tissue, number 3, muscle tissue, and number 4, nervous tissue. We have studied about epithelial tissue, which is squamous epithelium, cuboidal, columnar, this sort of epithelial tissue. Connective tissue, like blood, bone, cartilage, and connective tissue proper. And now we get to a stage to learn muscle tissue. And again, for the muscle, there are 3 types of muscle. They are skeletal muscle, cardiac muscle, and smooth muscles. So of course, we are just going to talk about skeletal muscles, which is the organ. Together with the bone, it will make a musculoskeletal system, which gives us a locomotor function. So it's a locomotor system. We are not going to talk about cardiac muscle or smooth muscle, which is for the gastrointestinal tract. What are the characteristics of skeletal muscles? So the most important thing is contractibility and excitability. Contractibility means when there is a stimulus like electrical or thermal, it will contract. Whether when the electric or chemical come and excite that muscle tissue, it will get excited and it will contract. And it has elasticity, being stretchable and return to the normal stage, and extendability. You can pull the muscle to some extent. It is extendable. Once you release the stress of pulling, the muscle will go back to its normal length. That is what we call extendable. So here I want to emphasize between elasticity and plasticity. Elasticity means when there is a stress, for example, like a tension pulling the muscle, once the tension has removed, when the stress is removed, it will go back to its normal shape, normal position, normal length. That is the quality of elasticity. When it comes to plasticity, that means you can put a stress on a tissue being stretched under pressure, and then when you remove the stress, that tissue will go back and get shortened, but it will not get back to its original position. That is what we call plasticity. So in muscle, it is not plasticity, it is elasticity. So which means if the muscle is cut, it will just retract. It has the contractility. Let's look at the next slide. This is a gent who jumped out of the window from third-story building. This is his left elbow. The bone popped through the skin in front of the elbow. Usually we will have a biceps muscle and tendon in front of the elbow. Now it is gone. It is contracted and retracted. This is a slide just trying to compare different cities. The top one is Paris. Lower bottom left is DC. Lower bottom right is London. Wherever they are in the same hierarchy, which means the same level, the capital city of different countries. So we talk about cells. We have talked about squamous cells. So cell plus many other cells will make a tissue. Tissue plus many other tissues will form an organ. Organs plus other organs will form a system. We know that. So where is the equivalent of squamous epithelial cell comparing to the muscle, skeletal muscle? It is the muscle fiber. So squamous cell plus squamous cells, many squamous cells, will make a squamous epithelial tissue. So muscle fiber plus many other muscle fibers will make a muscle fascicle. So the muscle fiber is equivalent to the squamous cell hierarchy level, because you can still divide further into many different parts for the muscle fibers. These are the fake cadavers showing the different muscles. These are the real cadavers exposing the muscle being removed, the skin. So this is a photograph of a Swiss train with the Swiss Alp background. Let's say if we think this train as a myofiber, in other words, if we take each an individual coach as sarcomere, which is the most basic functional unit for skeletal muscle. So one coach is one sarcomere. If we arrange all these sarcomere in a linear fashion, so one sarcomere, another sarcomere, and many other sarcomeres to follow. So if we arrange all these sarcomeres in linear fashion, that the whole train is called myofibrils. So myofibrils are made of sarcomeres, which are arranged in linear fashions. Inside the sarcomere or inside the coach of the train, there will be passengers, which is equivalent to the myofilaments, which are actin and myosins and some other proteins. So don't forget this picture. So a sarcomere is equivalent to a coach of a train, linear fashion arranged. It will be called myofibrils. Okay. So let's say we are seeing many trains, like 1, 2, 3 at least. So if one train is equivalent to the myofibril and multiple myofibrils together, which is wrapped with the sarcolemma. Sarcolemma is just like a cell membrane equivalent. And on top of that, you better loop around with the endomysium. Then you are going to call this structure as mast cell fibers. Mast cell fiber is equivalent to cell. So a cell has cytoplasm and cell membrane. So a mast cell fiber has sarcolemma and endomysium as a membrane, and inside there will be myofibrils. Myofibrils are made of sarcomere. So we will move on. These mast cell fibers group together, wrap around, enclosed with the perimysium, and it will become mast cell vesicle. Another similar fashion, mast cell vesicle being grouped together, wrapped with the epimysium, that will become the mast cell, which is an organ, which is something we can feel with our hands. A way to remember is you live in Sacramento, you take a rail, subway, and you go to ER, and you meet a person who is a fascist, but that fascist is quite muscular. So Sacramento is equivalent to sarcomere. Sarcomeres together become myofibril. These rails became fiber, group of mast cell fiber became vesicle, and these vesicles became mast cell. So this is the hierarchy we need to know. This will be asked in the exam. This will be asked for the undergraduate, for the postgraduate, and even when you do the board exam, the same question comes again. So let's look at the picture. So now we can see sarcomere. Let's start from the sarcomere. If we arrange sarcomeres in a linear fashion, one sarcomere here, another sarcomere here, another sarcomere will be here in a linear fashion, and you are going to call this structure as myofibrils. Myofibrils group together, packed with sarcolemma, and on top packed with endomysium, that will become mast cell fiber, which is cell equivalent comparing to epithelial tissue. So once you got a mast cell fiber inside, you're going to see a nucleus, endoplasmic reticulum. In bones, in muscle, endoplasmic reticulums are called sarcoplasmic reticulum. Okay, so group of myofibrils, move on. muscle fiber, fiber become physical, physical become muscle itself. I believe we all understand the hierarchy of a muscle structure. So the next step to know, remember, not to forget, is what is a sarcomere? A sarcomere is a basic functional unit of muscle. I'm not going to talk about this slide. I believe everyone knows by this time. So some terminology, like if we think about a cell have cell membrane, which is called cytoplasmic membrane. And inside the cytoplasmic membrane, the space is called cytoplasm. So equivalent cytoplasmic reticulum in master cell is called sarcoplasm. Cytoplasmic membrane in mast cell is called sarcolemma. Endoplasmic reticulum in cells, in the mast cell tissue, we name as sarcoplasmic reticulum. And sarcomere is equivalent to the osteon or neuron, or that is the basic functional unit of a tissue. And when we say that muscle is a form of tissue, those tissues are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. So muscle is a separate group. But in reality, a muscle is a composite tissue. So made of muscle fibers together with connective tissue, which are epimysium, endomysium, and perimysium. These are the fibrous connective tissue structures. So when a mast cell starts from one part of the body, which we call origin, then become big muscle fiber, muscle, and then it will go and insert or go and attach to another part of the body, which the site is called insertion. So when a muscle go and insert to a bone site, all these endomysium, epimysium, perimysium march together, make a group together, form with some fibrous tissue, and that is what we call tendon. So a tendon is nothing but a continuation of the endomysium, perimysium, epimysium, with some more fibrous tissue, go and attach to the bone and fuse with the periosteum, which is the covering of the bone. So there will be lots of blood vessels, which are very small, and we will call these blood vessels as capillaries. They are mesh and network inside the endomysium. So inside the endomysium, we get a lot of blood vessels, we will have a lot of nucleus, and we have a lot of sarcoplasmic reticulum. So we talk about origin and insertion. Where do we start? Where do we stop? And origin is the attachment of a muscle to a less movable bone. Insertion is opposite, attachment of muscle on a more movable bone. But it can be vice versa in some occasion. And I want you to think about that. And direct and indirect attachment to bone. So a muscle will become tendon when it's endo, peri, epimysium fuse together with more fibrous tissue, go to the bone and fuse with the periosteum. That is the indirect attachment. So let's say some of the fibers just go sharp deep down near end of the muscle, going straight down directly into the bone. It is direct attachment. But let's say if these fibers go and attach to the skin, the same structure, it's just go and attach to the skin rather than attaching to the bone, we will call that as raphe. Look at this person, he's about 35, he has been doing weight training, gradually improving, might use some steroid, And one day when he was in gym, when he was dropping down the heavy weight, and his right arm popped. He had it popped. He got pain on the right anterior elbow. And he noticed the structural changes in his right bicep muscle. If you look on the left side, the muscle bulk is normal. If you look on the right, there is an empty space like here, and the bicep muscle roll up and curl up because it contracts, because it loses its attachment to the radius. This is a case of distal bicep tendon rupture. So when we do an operation for this joint, we just make a transverse incision just in front of the elbow, try to fish out the bicep tendon. That is what we see, the bicep tendon, which is made of endo, peri, and epimysium with fibrous tissue. Again, this slide is just to show the origin and insertion of the muscle. So if you look at the muscle here, let's say vastus medialis, they will originate from the pelvis. When they contract, the femur will flex up to the tummy or chest. It's not when they contract, the body will not bend down. So this will just go upwards. So that is origin and insertion. I want you to take a break, try to absorb what I have learned. And after that, we go to next slide. Now we get to the microscopic anatomy of the muscle. As we say, all the muscles, big muscles, start with the sarcomere. So sarcomeres arrange in a linear fashion myofibril. Myofibrils group together muscle fiber. Muscle fibers group together muscle fascicle. Muscle fascicle group together muscle, which becomes an organ. So we are going to understand microscopic anatomy, which is the basic of muscle, that is sarcomere. These lines are important. If you look at a sarcomere, each and every sarcomere has a boundary. So one sarcomere start from Z line, go stop, finish on another opposite side Z line. So there, from here, there will be another sarcomere on this side. From here, there will be another sarcomere on this side. So the outer boundaries of the sarcomere is Z. Z is the end. It's easy to remember. So we are going to look what is happening in between two Z's. So let's look at this picture. So we have two Z's. Z's line or Z disc, they mark the boundary of a sarcomere. Inside, you will see the big fat myosin and small little thin actin. Actin is active and thin. Myosin, muscular, big fat. So they alternate, actin, myosin, actin, myosin, actin, myosin, actin. All these myosins, they are attached to each other with a protein, and these attachments under the microscope will be seen as a line, and that is what we call M line, middle line, in the most middle, that is M line. The myosins, they do not attach to Z line or Z disc. They only attach to Z line using these little yellow spring-like structures protein, which is called titin protein. Okay. And actins, they will attach to the Z line. Myosin, they do not attach directly to the Z line, they will go through titin protein. And when we look under the microscope, the site where we can see both actin and myosin, so this here. So in this central area, you will see both actin and myosin. So you are seeing all. That is what we call A. And here, you are not going to see myosin anymore from here, Z to myosin. So you will only see thin actin filaments with the titin. So it is small, very thin. We will name it as I-band. So in the center, around the M line, there is no actin, only myosin. So only myosin will be bigger than I. So I bigger become H. So that is the way you try to remember this microscopic structure of a sarcomere. So we have seen this already. We are going to understand again. So myofilaments will be, in a sarcomere, will be actin and myosin. Myofilaments start from one Z to other Z. Z is the end, that is the boundary of sarcomere. In the middle, myosin filaments are attached to each other with the protein, which under the microscope will be seen as a line, and that it is in the middle, therefore we will give it M line. Here, in this area, you are going to see both actin and myosin here, and therefore we give it a name A-band because all, everything. And here you will see only thin actin, very thin, and we give it I. In the center, we see bigger myosins only, and therefore it is bigger than I, so it is H. And don't forget, myosin doesn't attach to the Z disc. It will go only through via the titin filaments, titin proteins. Okay, so if you arrange these individual sarcomere in a linear fashion and make it a rod like a tray, that will become myofibrils, a group of myofibrils wrapped with the endomysium as sarcolemma and the endomysium. You are going to see a massive fiber. Inside, you will see nucleus. There will be sarcoplasmic reticulums. Let's look at the next slide. So inside the sarcolemma, you're going to see, we saw nucleus, we are going to see mitochondria, endoplasmic reticulums, or these T-tubules, which are the invagination of the sarcolemma in between the myofibrils and the sarcoplasmic reticulum invaginate together, they form a triad. So two sarcoplasmas on either side. In the center, there is T-tubule, which is invaginating in between all the myofibrils. And then on top of that, the sarcolemma wrap around all these structures. On top of that, there will be endoplasmic reticulum. So I'm going to repeat this again. A for all, both actin and myosin. Z, the end, the outer boundary of the sarcomere. M is in the middle, is a protein which is connecting the myosins in the most middle. I is very thin. This is just a T10 protein and actin. And H is twice I, and it is the myosin only area. T10 protein is runs from the Z disc or Z line to the thick filament. It keeps the myosin filament in place. It is a spring-like, coin-like structure, and it can be stretched, it can be relaxed. And don't forget transverse tubules and sarcoplasmic reticulum. Muscle contraction, basically there are two types of muscle contraction. They are concentric contraction and eccentric contraction. Concentric contraction means one, the muscle is generating force, at the same time, muscle gets shorter. That is concentric. Eccentric contraction means muscle is generating force, at the same time, it is getting stretched, muscle lengthen. That is eccentric contraction. I want you to take a break now. We have study 2, number 1, hierarchy of the muscle structure. Number 2, microscopic structure of the muscle. Because we are next slide, we are going to talk about how does a muscle contract. Let's begin the next slide. So we are seeing the muscle fibers, which is a group of myofibrils covered by sarcolemma and endomysium. Inside the sarcolemma and endoplasmic reticulum, you are going to see the nucleus, you're going to see the sarcoplasmic reticulum. So the spinal cord here, from the front of the spinal cord, our motor neurons come out, go and supply to the individual muscle fiber. A motor unit is a motor unit and the muscle fiber. So one motor neuron plus one muscle fiber, we see this group, this couple as a motor unit. So motor unit means muscle fiber, not myofibril, muscle fiber, and the neuron which directly supply to that muscle fiber. So that is a motor unit. If you go detail at the junction between where the nerve come and attach to the muscle, we will we say that is a neuromuscular junction because the neuron come and attach to the muscle, neuromuscular junction. So that's neuromuscular junction. This is the end of the nerve. This is the muscle fiber. Microscopically, there is a gap, which we will say is a cleft, and there is a synaptic cleft. Synapse mean two neurons or two structures meet together. There is a cleft. At the end of the motor neurons, they will have acetylcholine vesicles. When they get stimulated, the acetylcholine will come out from the end of the neuron into the synaptic cleft. So looking more detail at the synaptic cleft, this is the end of the motor neuron. There will be acetylcholine containing vesicles. When there is stimulus, this acetylcholine will come out into the synaptic cleft. that is a receptor for the acetylcholine. So acetylcholine will go and act on that receptor. With time, these acetylcholine will be degraded into choline and acetate. The enzyme which degrades acetylcholine is called acetylcholine esterase. So there are some conditions where we can have problem at the neuromuscular junction, like organophosphate poisoning, like fertilizers. They stop the acetylcholine esterase. Therefore, we are going to get loss of acetylcholine in the synaptic cleft, and that can give us problem. When we have botulinum toxin, it will stop releasing acetylcholine there. We will not have acetylcholine in the synaptic cleft. So botulinum toxin and organophosphate poisoning, they are opposite. One, organophosphate increase the acetylcholine in the synaptic cleft. Botulinum decrease the level of acetylcholine in the synaptic cleft. So let's say now we have enough stimulus from the spinal cord. It gets to the neuromuscular junction. Then it stimulates, acetylcholine come out. That acetylcholine will come and act on the receptor. And there will be signal changes, electrical changes in the muscle site via the receptor that stimulates the sarcoplasmic reticulum, and then that goes along the T-tubule, where because T-tubules contain a lot of calcium. Once this electrical signal or impulse got to the T-tubules will release calcium. So the calcium come out from the T-tubules, and then it will start reacting on the actin and myosin filament, which are myofibrils inside the sarcomere. So on the left side picture, it is a normal resting stage where you will see the balls, which are actins. There is a protein called tropomyosin, which is running on top of the actin. And there is another protein called troponin. So usually troponin is attached close to the tropomyosin, and tropomyosin attached to the sweet spot on the actin where the myosin come and attach when the muscle contract. So in a normal state, these myosin binding sweet spots on the actin are covered with tropomyosin. So when the calcium get to the actin myosin area, this calcium will bind to troponin. Once the calcium binds to the troponin, it will irritate or stimulate the nearby structure, which is tropomyosin. Then tropomyosin gets conformation changes. Because of that conformation changes, it moves away. By moving so, it will expose the myosin site, binding site on the actin. By that time, the myosin will be able to come and attach to actin. So with repeated attachment and relaxation, the muscle will contract. This process needs energy, which is ATP. So we have talked about neuromuscular junction. We have talked about motor unit. So look at this slide. In this slide, the top one, it is in a just 1 minute. Okay, let's go back to this slide. So if you look at the drawing, the sarcomere on the top, it is in a relaxed state. So two Z lines are wide apart. And if you look at the drawing in the bottom, the two Z lines are squeezed together. So being squeezed, the muscle is contracting, concentric contraction. So two Z lines getting close together, muscle get contracted. In a resting normal state, the T10 proteins are long and relaxed. And in a contraction state, T10 protein gets squeezed, two Z lines getting close together. And you will notice that the length of the actin or myosin hasn't changed. They just glide or slide on top of each other. That is what I want you to take from this slide. So when the muscle contract, when the sarcomere is in action, the length of the actin and myosin doesn't change. They just glide or slide on each other. The T10 protein here, the length will change. These are the websites which I find very useful. But have a look if you have time. Types of muscle fibers can be classified depending on how they make their energy or how fast they can contract. So those muscle fibers which use oxygen to get ATP, they will be called oxidative, oxygen-using muscle fibers. And those muscle fibers which generate ATP via glycolysis pathway will be called glycolytic fibers. If we classify depending on how fast they contract, which means how quickly they break down the ATP and generate energy, they will be either fast or slow fibers. Fast muscle fibers can be both oxidative or glycolytic, and slow muscle fibers are usually oxidative. If you look at this gent, he ran one of the fastest guys in human history. So his muscles in the lower limbs will be fast contracting fibers. And he is just a sprinter. He doesn't run marathon usually. And I believe his muscle fibers in the lower limb will be fast fiber, which is oxidative, past, short run, gone, 100. So there are a few clinical conditions which are associated with muscles. Number one is rhabdomyolysis. So this happens when a person is lying on the floor or being under pressure, one big muscle is under pressure. In these scenarios, like a slow muscle fibers, slow acting muscle fibers, which use myoglobin, which is equivalent to hemoglobin, because it is inside the muscle, so they are named myoglobin. And the long, prolonged pressure, these myoglobins break down and then get into the bloodstream and excreted in the urine. If the amount is too high, then the kidney lost its ability to excrete myoglobin. That is a condition called rhabdomyolysis. So that is for the slow contracting muscle, which is under pressure, and muscle became to get necrosis and lysis, and that's muscle breakdown. Exercise training, so we can train our muscles depending on what we like. So although we say there are slow or fast muscles, oxidative or glycolytic muscle fibers, in one muscle, it is combination of all, but of course, one group we dominate. So if we trying to train like the way we train our lower limb muscles, because usually, lower limb muscles will be contracting slower than upper limb muscle. It's for everyone, but for someone like Usain Bolt, the lower limb muscles are contracting really fast. How do we get it? He just train so that the lower limb muscle fibers become fast contracting muscles. If we lie in bed, if we do not do anything, if we do not use our muscle, they will go into atrophy, that is called disuse atrophy. And there is a medical condition where we say muscular dystrophy. I will explain that in later slide. And fibromyalgia, algia means pain, muscle pain, fibrous and muscle tissue pain. So the pain is all over the body. The cause is poorly understood. Let's go to the muscular dystrophy. One more slide. This is a condition called Duchenne muscular dystrophy. The cause of this is the absence of dystrophin protein in the body. It is usually happen in male only, and it is X-linked recessive. It usually starts at the age between 2 to 6. So when the child is starting to mobilize and walk around, and when he is trying to stand up, he cannot because his muscles are weak because of the muscle dystrophy. Why do they get muscle dystrophy? Because they do not have dystrophin protein in their muscle. So that is the way he is trying to stand up. So initially he's crawling on 4 limbs, then gradually use his arms, putting on his knee, try to stand up, and gradually he is able to do this. Okay, last slide. We saw this gentleman at the very first of the lecture. Now here he is. That is life. That is what we call sarcopenia. So with age, as the age incline, in old age, we are going to lose our muscle fibers. So we cannot stop losing muscle fibers, but we can do weight training and using the remaining fibers to get the same strength. Because muscle contribute a large portion of the body, which means it will contribute a large portion for the body weight. And therefore, when you get to old age, you're losing muscle and you're going to get weight loss. Androgen has positive effect on the muscles, but very sadly, skeletal muscles will never undergo mitosis. So once you lose it, you are not going to get it back. So to make it short, from today's lecture, what we need to know is number 1, hierarchy of the muscle structure. Number 2, microscopic structure of the muscle, which is sarcomere. And number 3, how does a muscle contract? And number 4, few limited clinical conditions. And don't forget sarcopenia, which we are going to have one day if we live long enough. I thank you for your attention. I'm going to stop here.